Hello, good afternoon, good evening, everyone, or good morning from wherever you are joining us. Thank you so much for joining us for the second session in the series that we are currently having on TB genomics. For those of you who joined us on the first webinar where we had Dr. Francis Chikuse from the Africa CDC, will set the scene so perfectly for us in terms of the sequencing capacity that is available on the continent. So today we are continuing with that series, and we have Anita Suresh from the Foundation of Innovative and New Diagnostics, who is going to be continuing to talk to us about the evidence that generated data for the WHO recommendation. Many of you will be aware that WHO in 2023 gave the recommendation for targeted next generation sequencing to be used for diagnosis of drug-resistant TB. This is an area that we, as I was saying again, just giving an overview of the aim of this series that we are having on targeted next generation sequencing, and saying that WHO has given this recommendation that can add to the toolkit that we currently have for drug-resistant TB. So we're really trying to help countries learn from each other, learn the best practices for implementing, and learn some of the strategies on how they can begin thinking about introducing targeted next-generation sequencing. So before we begin the webinar, just a few housekeeping issues. This webinar is being recorded. You can choose which channel you want to be in. As you heard, we have Portuguese and French translation available. So you can go on the globe icon within the Zoom toolbar, and you can choose which channel you want to be in so you can follow along in the webinar. We also currently have a poll that's running to help us to understand how targeted next generation sequencing for TB genomics is currently being used in your countries. So if you could help us by answering that poll, that would really guide the discussions that we are going to have during this session. You can put your questions in the Q&A box, and we will respond to the questions live. And also we have Swapna from the genomics unit at FIND who will also help with responding to some of the questions. And if we fail to get through all the questions within this webinar, then we will have Anita and the team respond to the remaining questions, and we will send those questions along with the video of this webinar, as well as the slides from the presentation. Those will also be shared with you. You will also receive a link to download your certificate of attendance should you need to. So with that, I will introduce our speaker, and we're really excited that we have Anita Suresh, who is currently the director of the genomics unit at FIND in Geneva. And she is really she's the go-to person right now if we want to know anything about genomic sequencing for TB, and not only for TB, for AMR and pandemic preparedness. She was the PI on the Seq Treat grant that was funded by UNITAID, and that's the project that provided the evidence that WHO used to write up this recommendation for targeted next generation sequencing. She has led the efforts to generate this evidence, and she has over 20 years of experience across the entire biomedical value chain, from commercialization of diagnostics, technological assessments, and research and development. She has an MBA and an MSc in bioinformatics. So we are really quite excited that she's going to be sharing her expertise with us and helping us to start thinking where we can place targeted next generation sequencing within our TB programs. So over to you, Anita, and thank you so much for joining us.
Thank you.
Stop sharing.
Yes, that's okay. Thank you, Lucy, for that very kind introduction. Good morning, good afternoon, good evening, bonjour, bon dia to everyone. And thank you, Lucy, Collins, and Francis, and the ASLM team for having me on this on this platform, because this is, I think you'll have been doing a wonderful series of all the ECHO LabCoP talks. So it's wonderful to get an opportunity to be part of this, to share a little bit about also the Seq Treat and the tNGS experience we've had, and also to see, if there's things that we can help as you guys, as all the countries and your partners, your network is starting to roll out or strengthen your own sequencing in your countries for various applications, as we saw from the poll itself. So again, as I understand, all of you pretty much are the TB experts. You are the ones working in the field. You are in the labs, you're in the programs, you're in the pretty much in the community, making a difference and implementing diagnostics and care for everyone affected by TB. So this is all numbers and stats you're more than familiar with, but it's always important to start with why we're doing this. And very much as again, literally saying things that but the fact that TB is treatable, curable, yet it remains the leading cause of death from an infectious disease. Still, despite COVID and everything else, this is the biggest one. We still have over 10 million people that fall ill with TB every year, and even more than 1.25 million of people that died of TB in 2023. And close to 500,000 people develop drug-resistant TB, which makes it so much worse in terms of treatment and management and, of course, transmission and so on. So, of course, universal access to comprehensive resistant testing is the one that's needed. We know that today that less than 50% of drug-resistant TB patients get diagnosed. This number has definitely increased over the years, but it's still not where we want to be, not yet where we need to be. So early detection and optimal treatment is very critical. So we need to get people on the right treatment from the start. The good thing is, of course, there are new drugs, shorter regimens available with the BPAL, BPAL-M, and so on. All of the newer drug combinations regimens that have been tested and proven to work effectively. But it's important then that as we roll out and scale up these, the availability of these drugs and regimens, we need to ensure these drugs continue to remain useful for as many people as possible for as long as possible. And hence, we need to have ways to do the drug resistance testing alongside the treatment rollout to ensure that, again, as many people have access to the good treatment that actually helps and it stays efficacious in the market, in the community for as long as possible. So as we said a little bit on, again, this might be basic information for many of you, so bear with me just to set the ground here in terms of targeted sequencing. Being able to do the DNA extraction, the enrichment for the targeting, or the enrichment of the specific targets within the genome, as you see here, followed by sequencing. So you are trying to interpret specific variants from the amplicons to be able to predict resistance phenotypically. This is because TB has a highly conserved genome, and there is a lot of knowledge accumulated over the years, continues to accumulate, which we'll speak to later, on what resistance is encoded by the different mutations. So we were able to reliably predict resistance from mutations. So we don't have to sequence the entire genome, but really go to these most important regions and be able to still take action on these results. Now, whole genome sequencing, of course, has a very important place, and then in research, discovery, but really from a surveillance perspective. Today, it still requires starting from culture. And this is where you're extracting DNA, because now that you've cultured up and grown up the bug, you pretty much have an excess of the bacteria. Then it's so much easier to extract DNA from that culture, do the same fragmentation library prep and sequence, and now you're getting all of the information, the entire genome. This is obviously useful because you might be looking for things that are not yet common in the world. There might be emerging resistance. You might be doing it for transmission mapping. You might be doing it for outbreak outbreak response, outbreak detection of certain resistance in certain regions, and so on. So there's a lot of value to whole genome sequencing as well. But from a programmatic clinical standpoint, especially if you're talking about as quick as possible a result to be able to guide the clinician, to be able to treat the person affected with TB, you do want to be able to go direct from sample. And this is what targeted sequencing allows you to do. Hence, our focus pretty much for Seq and Treat was on looking at targeted sequencing starting from the sputum sample for the use in drug-resistant TB diagnosis. So breaking this up, now I've described the method, but really why is this important, right? We talk about targeted sequencing, why this is a good solution. I think the time, obviously the time to result is one of the biggest factors here. So of course, culture is comprehensive, phenotypic, if you're able to do all of the phenotypic DST, drug susceptibility testing for all of the drugs that are available and relevant to your region or country or setting. But it does take 6 to 8 weeks, as you well know. So that is really not a diagnostic by any means if you're going to take this many weeks. Yes, you're starting to put people on a treatment, but then you have to adjust it. There might be responses. You might also be, unfortunately, starting on the wrong treatment and so on. So there's a lot of challenges, and we want to be moving away from something that takes this long, saying, how can we make this faster? Now, what you don't see in this picture is the PCR test. Obviously, PCR test is very important in terms of the rapid detection of TB and potentially also detection of at least the RIF and INH resistance, at least the first 1 or 2 drugs, to ensure that, hey, this is maybe susceptible TB, so I don't need to worry about drug resistance, so I can start with the standard first-line regimens to be able to help the patient. But we're talking now about one step after that. We're talking about how can I make sure I'm getting, again, all of the right combination of drugs for patients at the right time and for the early enough for it to make a difference. And that's where you want still the comprehensive ability of phenotypic DST with the rapidness of a molecular test, because again, you're able to do the PCR, do the sequencing rapidly, and be able to generate the result very quickly to get back to the clinician and hence get back to the patient with the result. So you'll see the difference of 6 to 8 weeks, potentially over 2 to 3 weeks with whole genome sequencing, since we still start from culture. And with targeted sequencing, we are targeting a time frame of 1 to 2 days. Right now, it was still 2 to 3 days at the start of the project 6 years ago. And I think we did say already some of these aspects of why targeted sequencing. You've had other sequencing talks in this webinar series that spoke to this. Nevertheless, again, highlighting some of the importance when it comes to TB. Of course, we did say the strong genotype to phenotype correlations, so we can confidently predict resistance from mutations. Overall, a sequencing as a technology is high throughput, any throughput really. So you could have low throughput, but it can also scale up to as much throughput as you need and over time as you want to grow it. Lower by safety requirements compared to a culture lab. It's also faster results really, because we set the turnaround time itself to inform clinical decision making. Another most important thing now when you compare to molecular is how adaptable sequencing is to new genes and new mutations. To basically, I don't have to go back to the drawing board to build a brand new cartridge or assay to update the test. I can still add maybe some tweaks. I can make some changes in the software, or I can make some changes in the amplification in terms of the primers, the targets in there, to be able to update the test according to as knowledge base increases. More on this as we come to the mutations catalog section. And of course, one of the biggest value adds of sequencing, of course, a multi-disease platform. You've already seen it in many of your countries, been used actively during the pandemic, potentially using it also for malaria, for HIV, and so on. So this is where you want technologies that can be used for multiple health concerns and multiple health priorities. So really, this targeted sequencing presents a real paradigm shift when it comes to diagnosis of drug-resistant TB direct from samples. So I've said all this, and there's a reason now why we went on to Seek and Treat, and why securing that funding from United was important when we started, which was really 2019. Because at that time, there was not enough evidence, really. And also at that point, capacity was still at a very early stage, nascent stage, if you will, across the world. So the goal of Seek and Treat was really to generate evidence and catalyze in-country capacity to support the global adoption of end-to-end targeted sequencing solutions for comprehensive drug diagnosis of drug-resistant TB. Now, it consisted of, obviously, with multi-year projects, so many different parts to it. I'll describe some of these in more detail, and of course, talk to every one of these phases. So the first phase was definitely most important, rather the foundation, the fundamental goal of Seek and Treat was to generate that evidence. So really consists of two phases: analytical validation and then clinical evaluation in the field in countries. In parallel was the idea of generating the mutations catalog and the TB knowledge base. So this was that whole evidence base needed to say that, hey, I can confidently predict resistance from mutations. Until then, we only had, there was enough data, or there were enough tests that were basically had all the information for RIF and for INH for the most part. But otherwise, for the rest of it, while people knew of these were more things that companies did their own research or looked up, did literature reviews and so on. There was no existing sort of an endorsed list of mutations or a list of truth of high confidence mutations associated with the resistance. So this was an important goal of the project, to be able to also generate that, not only for this trial, not only for the products that we are evaluating, but in general for the TB community as more tests are developed in the future. Both of these aspects were essentially to inform WHO policy on the use of tNGS. Obviously, one step beyond that has been we can't just stop there, because with sequencing, I can't just tell you, okay, we've gotten the evidence, products are available, now everyone go ahead and use it. It is still a workflow, not a box today. So you still had the ability or the need to assess some of these implementation models. What are some different ways? What are those challenges when you actually operationalize sequencing in the program, in the field? And really some of the other tools required to help prepare for scale, the implementation tools, the planning tools, the budgeting tools, and so on. So this was a chance to bring all of that together and pull it out here. Jumping quickly to the analytical clinical evaluation itself. So when I say the analytical validation, it is really a lab validation, if you will, saying, does the product do what it says, what they claim they can do? Literally, can they call those mutations that they have said they can? So this was essentially the goal here, was to say, is the test reproducible, and whether there's accuracy in terms of the SNP calling, the mutations calling. And from these results, it is clear when a SNP is present, it is called accurately. So with sequencing, a big part of it is, if there is a result, if there's a mutation, it'll find it. You won't get a wrong result. You could get no results if there's not enough load. But again, more on that later as a challenge. The phase 2, again, the next important part, was essentially a multi-center diagnostic accuracy study across 3 countries, South Africa, Georgia, and India, and a trial population of confirmed pulmonary TB patients at risk or proven to have DRTB. So a lot of details in this. The good thing is this is now published. So the objective was really to look at the diagnostic accuracy for resistance for multiple tests to 13 anti-TB drugs at the time. Now, in terms of the platforms that we evaluated, obviously at the time, the idea was we did a call for what products are available that have an end-to-end workflow for DRTB detection, starting from sputum sample. So at the time, we had about 3 products. So I'm showing you details of 2 of these here, because these were the ones that fed into the evidence package that was shared with WHO. So the first one is the GenoScreen Illumina combination, and the second one is the Oxford Nanopore product offering. And you will see, again, the components are pretty similar, right? So you'll have, starting from the sample, the DNA extraction, the target amplification, which is a PCR step, the library preparation itself, so that the sample is essentially ready for sequencing. You're then pooling, quantifying all of those. So all this QC that you need to do before putting it on the sequencer, sequencing itself, followed by the bioinformatics, the analysis, and the report of the results. These specifics on what they are, you can see later. Obviously, there are different combinations of this, and the point of this slide is really to highlight that the goal was to say whatever the manufacturer in a way says, this is the start to finish, the end-to-end solution that we are presenting to evaluate is what we used for this study. So jumping many steps, many months, and many probably years forward, because this happened during the COVID pandemic. The clinical results have been published. So the whole idea was that the study was completed. We have the results of this. It was very clear that we could assess the diagnostic accuracy and, more important, the failure rates across various drugs, because that's an important aspect of diagnostic sets. How often does it not work? Because that gives you an idea of what are there things you can optimize further to ensure that essentially lower that failure rate. So it was clear that from this study that there was a potential of tNGS to replace conventional methods by providing accurate and comprehensive drug resistance profiles directly from samples. I'm going to do a quick summary of results. It's been over a year since it was published. So it's, again, things you would have seen and heard enough. Essentially, we saw that both solutions provided accurate and reliable, reproducible results from direct clinical samples. More than 95% of the samples from both produced TB sequence data. There was, of course, since we said the high rate, you're seeing some sequence failure. So if you see this chart, it's really about where you got complete sequence results. You got partial, where there was some targets were missing, as in some resistance data, there was just not enough information to make a call. Or there was complete failure. There was no sequencing result. Sequencing was not possible, or it failed. It basically did not give results. Now you see, of course, you can see the highest ones are complete. As we said, more than 95% of these produce the results. Now, sequence failure, where you do see it, you see it's essentially associated when there is very low bacterial load. How do we say this? Is because we are using the expert semi-quantitative results as a proxy. So what you see here in terms of the gene expert results, because this was obviously the first test we did, and in terms of an inclusion criteria. So you see, depending on whether the expert semi-quantitative was a high or a medium, you're seeing pretty much complete results coming there. Once you start to go to a low and very low, you're seeing a lot of dropout happening. So I think that was an important aspect to say, that sequence failure is definitely associated with lower bacterial loads. And in this case, we saw GenoScreen had more than 91% of sediment samples produce information, and ONT more than 94% produced the results, DR results. So this is obviously, again, at the time of our study, the companies, this is useful information that obviously companies have been utilizing to improve on their workflow, to improve their sensitivity, essentially, to be able to either enrich, be able to amplify better, to be able to get more DNA, or to do more with that DNA, to amplify it better from the extracted DNA, and so on. So there's various ways to tweak this, but the goal here was to just see how it worked as a product. More details on the GenoScreen accuracy. So again, you're seeing very good results in terms of sensitivity, and they really met the target product profile minimum, the published target product profile for NGS, for drug-resistant TB detection, not just for NGS, but really for any molecular test for drug-resistant TB detection, published the WHO TPP. This test did meet all of those. Linezolid was the one where it was definitely lower. But from this, can at least understand that the performance, as the knowledge of mutations associated with resistance is expanded, there's potential that this can move up. This can be increased, essentially. So now for Oxford Nanopore, again, you're seeing good results for all of these, the first set of drugs, even some of the second-line drugs, but you're seeing that dropout in the newer drugs, the newer Says drugs. So again, this here seems like the sequence data was generated. What they did not have is an interpretation or results reported for these drug targets. So this will become relevant as I talk about the drug targets and I talk about the mutations catalog. So jumping from there, all of that evidence, again, jumping several months now to say from evidence to policy on NGS for patient care. So this was all sort of in the hands of WHO that was shared, and then the whole guidelines development group that was convened to look at evidence. I think about over 80% of the data came from Seq Treat. There was, of course, other data from other studies around the world that were submitted. So it was really a sum total of looking systematically across all of that evidence to be able to review, assess, and see what it means. It was great to see the final guidelines that were issued last year to show there was class-based recommendations on the use of tNGS for DRTB diagnosis. So this was done both for starting with people with bacteriology confirmed pulmonary TB itself, and then for those with confirmed RIF-resistant TB. So in both cases, in this case, the first one is direct just for those who already are known as TB positive, and the second is for those reflexed from a RIF resistance test. The products evaluated, as I said, the T-plex MIC-TB, which is a Genostrain Illumina combination, and the Oxford Nanopores Ampor TB test, as it was called at the time, and one more test from Shengting in China called TB-Seq. And you can see the results was really for different drugs for different. So each test was really analyzed based on the performance per drug. And that is important because the idea is that over time, this can be improved. So certain drugs, it's a default, like it's table stakes. You have to make sure the test performs and gives the resistance information for these important drugs. Now, additional newer drugs, there is understanding that not all the knowledge even exists today. Not all the mutations are proven to be associated today, are known today. But over time, as this knowledge increases, this should and will translate to better tests or increasing the performance of these tests for these drugs. So moving there from now, we said about that was, okay, so there was evidence and policy. And obviously now from policy to implementation, there's a lot of aspects here, right? Things that need to be done. While people did say, yes, it's comprehensive, it is reasonably convenient, it's still a workflow, but it's pretty, it's something that one can be trained to do and be able to follow. There's SOPs for it. And it's adaptable, rapid, so on. There's obviously still challenges. There is setup. There's complexity of setup itself, there's specialized infrastructure expertise required, all of those that you see in any standard sequencing presentations really, or from experiences from countries. So I think the whole idea was, you can see there was a little cost effectiveness study done as part of the WHO work. The idea is obviously it is cost effective. So tNGS is cost effective, really depends on the context, and it is dependent on things like prevalence of the resistance. On the current standard of care, is there PTSD being done today for all of the tests? Is there some tests that nothing is being done for some resistance marker, so we don't even know if we are blind to that information, in which case this is truly going to add value and actually give you all those results in one test. Obviously, then how it's being implemented, is there a centralized facility where sequencing is being done already, and you can do more TB sequencing there, or do you need to set up multiple centers from the start, which means obviously upfront investment in that. So while there was at least it was acknowledged that it is acceptable, implementable, there is some inherent complexity, and the fact that we still need to investigate further implementation models and different algorithms because they will be dependent on the context. So as I said, one of the things at least was highlighted in the operational handbook is about where does tNGS fit in the DRTB testing algorithm. Now, obviously, depends on, again, the country setting where it's being used, and the burden of drug resistance as well. But really that is where the starting idea of where it can be, right? There's obviously different models that can be used, but the idea of where, because you're getting results for these drugs that you don't get anywhere else and may not be using, and it'll take too long to get it from culture, at least this serves as a way to get those results first. And if there's a resistance risk or you're seeing resistance, at least you can modify the treatment or review treatment accordingly, even as you wait for TB culture results. And in certain cases, when there's obviously, if you have XDR, if you have other tests that can give you a lot more information that you're doing as part of your routine programmatic testing, then where does this, what is the additional information you'll get from sequencing? And that's where sort of that decision would need to be taken in terms of how much more value am I getting out of this, and hence how many more, which of my sort of population should be prioritized for this at this stage. Ideally, everyone should have access to this test, but if not, where would it help the most for the clinician and for the program to help patients? I spoke a little bit on this translating mutations to resistance. Bear in mind, this can get a little nerdy, so I apologize in advance if it bores some of you. But we just, this is an important aspect because we're talking about how we know that now WHO endorses tNGS, direct from sample resistance profiling across these 10 plus drugs. Do we also know that what we're seeing is that, as I said, it's a molecular test. So what tNGS is identifying is mutations. Now, the interpretation is what tells us that these mutations are associated or predict phenotypic resistance, and hence you can do something actionable in terms of a treatment decision. Now, if you are not talking about every mutation ever, because not all mutations confer resistance, as you know from your genetics 101 classes, some are just have no effect, they're just benign, some are uncertain, others could be just level of resistance, not by itself, meaning it's resistant or not, but saying, okay, maybe I can still have treatment at a certain dosage and it'll still be effective. So this interpretation is what becomes important, right, to effective care. And that's what makes also TB care challenging, because it is not sort of just this one treatment, this cure, this is not so direct one-to-one relationship. So we do require some standardized tools and guidance to say, how do we do this? How do we interpret, especially from a test? As we get more and more information, all of this big data that's coming out, you definitely want a way to say, to kind of harness that to say, all of this data means these 5 things or these 3 things. That's all you're looking. That's what at the end of the day you want as a clinician and as a program. And hence that was the purpose of that mutations catalog, right? So saying, yes, each of us can do our research. We can look at papers and find different ways. They could know this is important, or we'll have to prove why it's important. Really, can we have a standardized list of these? So it makes it easier for the most part, at least the most common, well-known mutations. It is clear that they are associated, and hence any tests that are made or interpretations that are made should at the minimum include these targets. So a lot of words here, but more important to say, if you're familiar with the mutations catalog, you know the idea was there was a version 1 released in 2021. The whole idea was, let us get as big a data set as possible of MTB data, so phenotypic and genotypic correlated data, to be able to say which are these mutations of highest frequency, which are associated with resistance, phenotypic resistance, and hence which are the ones that pop up the most, which are the ones that maybe pop up in some places or in some countries or in some instances. Are these important? Can we put them here, and so on? So different ways of classifying them. But to do this, I need as much data as one can have from the world. So you will see a version 1 had about 38,000 data sets. In version 2, this increased by 15,000 samples. Really, idea was increasing coverage. So many countries from their surveillance data, from their sequencing projects, contributed to this. And that's how we were able to add mutations and resistance information for bedaquiline and for linezolid, for delaminate, for clofazimine. Now, the version 3 that the team is working on very intensely right now adds another almost 30,000 data sets to this repository. So which means there's now even more information. And here we are now seeing mutations associated also with predominant resistance. Now, I think that that is important. This is going to take some time. It's never going to be sort of a one complete static mutation catalog, but it's how often do we need to update it to get enough for it to be useful to actually make a difference, both in improving a current product, but also in improving in a way the care around TB itself. Like how do we look at what is needed and the advocacy that's needed to say, okay, this we know more about these resistance, about these drugs. Let's make sure there is testing available and accessible that folks can actually get also the testing alongside getting access to better treatment. So all of this data sets that I'm saying where we are seeing this, of course, we are grateful to countries that have been sharing all of this data over time. And really over the last few months, it's been a lot of this, especially countries that have started rolling out the BPAL regimens, but they do have access, or they at least talk about they are able to do the phenotypic DSD and or have access to sequencing to be able to do those associated for those samples. There've been a lot of essentially contributors from across the world, which is what powers this. At the end of the day, it is data from, it is really information that you're seeing in the field that should feed back, that should allow us to say that, okay, now we can say with higher confidence, because of so much and all of this from the world, that this represents as close as we can what is happening in the real world. And hence, this is something you can be confident of using to be able for your programmatic needs. If anyone has seen the catalog itself, you will know that it's very, very complex. You will know that it's huge. First, there's a lot of things in there that makes it that don't make it as easy for, in a way, even a scientist, to look at, other than maybe a bioinformatician and so on. But there's a lot of information in there. So one of the things we have just completed, and hopefully will be coming your way soon, is a simplified catalog user guide. And the idea is, how do we simplify these, show these mutation tables so that you're only seeing the final grading. If you really want to know how it was done and why it was done, look at the entire catalog. But if you just kind of want to know, hey, tell me the bottom line, tell me what I need to know to be able to do something with this, this is your go-to guide. So some of the explanations for the grading are there. Some of the rules that are described there are decoded with some examples. So to really allow, again, people who are interested may potentially in utilizing it, or even updating tests or trying to develop new tests, this will provide a way to actually, a very accessible way to get to understanding the catalog of mutations and the value of those. And here's, again, don't worry about table. I've highlighted things that you need to see. But essentially, when we said, hey, the performance of tests can be improved as you get more information or more insights into the mutations associated with resistance. And so this is an example of just looking at the catalog itself. If we said version 1, version 2, why do I keep updating it? Because I want to improve in sensitivity, because I'll have more data sets to strengthen more frequency of mutations that are captured, more numbers. Just statistically, I'm going to have a larger base to be able to get this data from. So you'll see already the difference where we had pretty much nothing for clofazimine and bedaquiline catalog in the first edition. We were starting to see over 70, 78%, 74% sensitivity in the second edition of the catalog for these two drugs. And for linezolid, also an improvement. You're going to still see linezolid is still at this period. I don't know where it's at in the third edition. I hope it's increased a bit, but linezolid overall, as you know, is a very, very low prevalence resistance that you see in the world. So which means you're going to have very little data sets to contribute. This is a good thing. We do not want high prevalence mutation. We do not want high prevalence resistance as much as possible. So if something is low prevalence, that also means then we're going to have very little examples of it in the field or in surveillance to be able to power this the catalog or to power that knowledge base that feeds into the catalog. But that's where you're going to see that gap. You're also going to see very little, for example, from a phenotypic standpoint for linezolid. Many in countries, they may not even do linezolid testing for PTSD, and there's still challenges with that in terms of operationalizing phenotypic. So that one is still something that's low that you'll see you're only going to see incremental increases there. But for bedaquiline, clofazimine, you're seeing that significant increase as we add these mutations into the database. So I'm almost towards the end here, meaning that we have a few minutes here. So really, we spoke about the bulk of it, right? The core of it. What is that evidence that went in to inform WHO guidance, both from a use of tNGS, from a diagnostic guidelines perspective, but also for the mutations catalog in a way to say how do we interpret these mutation, the sequence changes of the sequence information, what does it mean in terms of phenotypic resistance, and hence what does it mean for me to actionably change or even recommend treatment for different drug-resistant profiles. Now, beyond that, that's great. So we have evidence, it works. There's data backing it. There's policy now. But then what are those practical considerations? So this is where I think I want to speak a little bit of. So we did ourselves some of the, just sort of wrapping up, some of the implementation pilots with countries that were already embarking on this or keen on doing implementing sequencing, at least to pilot it for potentially programmatic usage. So one was in Brazil, one was Indonesia, the third has been South Africa. So this has generated information, lots of the similar challenges that I think you've been seeing potentially in your field, not just for TB, but for other areas, all the way from supply chain procurement, access to these tests in a reasonable time within the countries, but also things like just the workflow, setting up like extraction, things that can be variable, unfortunately, things that are considered as a good thing by the manufacturer as, hey, it's flexible, you can use whatever you want, also means it's no longer standardized. So that means there's going to be options. That means there's still going to be some time and effort to optimize it or even to get standardized that workflow with that extraction system. So these are, again, practical things we're seeing in the field. So I think from our side, the project at this last stage of our project is say, what are some of the actually tangible products we can bring to the TB community? So you'll see some of these would include like technical operational guidance for the uptake of sequencing. So we have one, again, all of this work is with the WHO, GLI, and other partners. Seq Treat gave us, in a way, a mechanism to be able to do that together with these partners who anyway are the ones driving a lot of this work. So here it was essentially to say, okay, what is a manual, if you will, or what is some technical considerations that are operational considerations that countries should have in mind when they think of implementing sequencing, even for surveillance. And then the latest one right now is obviously for diagnostic use. But from both aspects, wherever you start, where most countries are going to start with surveillance, at least what are the things you need to start off so that you can be set up for success right from the start and not have it just as a research project, which comes in with a grant and then is done. You really want something that makes sense for your country and your setting that you can continue to sustain and actually provides useful, actionable information to your programs and ministries of health and really ultimately the TB community and patients. So lots of training aspects. I know ASLM we've actually partnered with ASLM on trainings in the past. I'd love to see if there's ways we can also help on the TB side if there's anything there, but I know there's a lot already good work being done by our implementers in the field. And then the inclusion of the TB NGS products in the global procurement list has been an important aspect, right? So again, these products, this is not necessarily there is cost associated with each of these components. A lot of these are available in the country today, but how do we make sure people can actually, or the TB program in a way, or the TB samples can actually access this infrastructure? So one of those was obviously ensuring, firstly, these products, the kits themselves are on the procurement list. Two of the big changes that happen in the field, or I think, good things that happened for the community, I should say, is this whole partnership of GenoScreen with Illumina and of ONT with bioMérieux. both of these partnerships are important because they're essentially tying someone who has either an assay advantage, a comparative knowledge and expertise and experience, or someone with a product, low sort of a low footprint or a small scale instrument with very highly scalable instrument with someone that has the assay knowledge and also the market presence. So I think both of these are very promising and really have a lot of benefit for the TB space specifically, I think initially, and then beyond. This is obviously valuable for the companies themselves because it gives them a much bigger, wider net that they can cast, but hopefully this means they can serve you better in the field going forward. Not just as a grant or research projects, but really for programmatic scale up. There's also all this, as I said, some little bit I spoke on cost effectiveness, but you're going to see some work coming out over the next few months as well on additional studies. This is something, obviously, it is clear that the questions that are coming are more on costing. It is clear that, I mean, you're seeing the benefit. When you get the results, it's beneficial. You know there's a reason you want to do this. By the time you embark on using sequencing, the program has seen that need or seen that gap that this is filling. But most important, the costing still makes a difference over the long run. Even if you have access to instruments or repurposing or leveraging existing instruments, you still need the ability to pay for the reagents over time. And that's where I think some of this manufacturer engagement and shaping are important. Groups like CHAI and others are working on these actively, really taking on from where Seq Treat kept it to the next step, right? Really making sure this really goes all the way to it benefits the maximum number of people. One thing I'll mention here is a genomics costing tool, something we worked alongside WHO, APHL, the UKHSA, the Global Fund, and others. And this is one where now TB and other pathogens have been added in as well. This is an important tool where it really can be used for country planning, forecasting, at least understanding what those costs are. So because a lot of the time it's about what are those different components, and we shouldn't just be thinking of the sequencer and the kits, but really everything else that's required to effectively run it and get a result that is usable. In like this concluding step in terms of expected improvements in the workflow, there's a lot you can think of. I'm sure you already know of a lot of gaps in the field or people working on this, or maybe you're working on some of these, which is excellent. So first one we said, when I'm really looking at now that'll happen, is really the updated mutations catalog, right? Reanalysis, and then being able to reanalyze some of these tests. If they made updates just in the software, you can envision saying, hey, I just have to rerun on the same data set with the new software, and let's see that improvement. Now, this was what we did earlier in the year, and this was data we submitted to the WHO TAG review earlier this year. So that is something I would say stay tuned for that. We should be seeing some communication from WHO soon on that. It was great that this is one of those instances where we've been able to see within a year of a new class of diagnostics actually recommended, in this case, targeted sequencing. WHO then convened a technical group to say that, hey, this is one test where we did say it can improve quite rapidly as increased knowledge happens. As long as the manufacturers act rapidly and make those changes, and there's a way to evaluate those changes, we will bring together the experts to actually review this data to say, hey, did they do better now? Have they improved? Which was the goal of that TAG meeting. I'm not speaking on behalf of WHO for sure, so just saying that this is something to look out for. But that's what, again, updates to the catalog gives that ability and that opportunity for improvement in performance of these tests. So taking that in the future, really, there's other versions. There could be new gene targets that are not in any assays today, but we need to be adding. So that also over time, as we get more information, especially phenotypic DST data in the field, that's going to be important to build this part of the knowledge base as well. And I cannot not mention AI, because there is a generative AI, essentially. You can imagine just using it to interpret the catalog. Just imagine you being able to ask, of course, a very customized bot to be able to say, I'm seeing this and this resistance, but I'm not seeing this. What does this mean in terms of interpretation? What does it mean in terms of treatment? You don't want this being done willy-nilly, but there is a potential for this, as you can see. You've seen it in all other settings. Definitely, there's a room and space and a potential for this within this field as well. At a minimum, to be able to interpret what is already known from a very complex catalog, but really at the next level, being able to help with some of the predictions from using machine learning and so on. So there's a lot of not just generative AI, but also machine learning network capabilities, regression analysis to say, how can we rapidly update resistance profiles in that knowledge base and be able to confidently predict all these mutations from resistance as more targets, as we get more information, because we may not get as many data sets like the 80,000 that we have to date. There's going to be much smaller numbers potentially over the few years, but we still want to do maximum what we can with that small amount of data. And then the last part is more all the other things we all ever wanted to see in terms of sequencing improvement. But I've only written things where I know somebody or the other is funded to do the work and continues to be funded. So it's like things of increased level of automation. Obviously, there's a balance. More automation could mean more costs, but at the same time, if it means over the long run, it does something even more standardized, that is a cost. I think as programs have made decisions for different areas, might make sense as a trade-off to at least consider. Then simplified extraction and processing. We could all do with simpler extractions. All this was done, the innovation was done during COVID. Absolutely, TB needs to have this advantage as well. TB needs to be able to do this. There's enough groups working on using swabs and other methods, other non-invasive sample types and so on. So all of those, how do we do the simplified extraction? But even from sputum, just making extraction easier, simpler, and better yielding. Library prep itself, it's still a lot of steps. It's still a lot of pipetting. You do want abilities to reduce the number of steps here. While we're at it, why not combine sample and library prep? Again, this is existing grants actually that are working on this effort, because there are cartridges that have been made for PCR. Why can't that also allow you to do library prep? Combine that so that what you're getting is something, a completely ready sample, processed and amplified, ready for sequencing. Of course, the holy grail has always been WGS direct from samples. If you can get whole genome sequencing direct from sputum samples or any other sample types, really, that is really where we want to be at, because we'll be able to interpret it, we'll be able to get all the information we ever needed, and more important, know what to do with it, how to do with it, and what we can do to make a difference. With that, I'll stop. As we said, some of this improvement aspect, but happy to speak to this later, where we did see that improvement just from an example of the mutations catalog for ONT, for example. Thank you so much. I wanted to, of course, thank our donors, Unitate, but everyone else that really has helped find possible. All the country technical partners we've had, the list is huge, so I won't try to name and forget some people that way. So everyone that's worked with us, and of course the whole fine sequencing team. Thank you, and happy to take more questions. I see the lot that came up. Sorry if I hope Swapna and others got to them, but I'd be happy to answer any others right now, Lucy.
Thank you so much, Anita. That was a brilliant presentation, and I'm seeing a lot of questions in the chat. Some people asked if we could rerun the poll we had at the beginning. So Fitsam, if you could assist with that for people who had not responded, how they are using tNGS for TB in their countries. I see a few questions, and Swapna, thank you so much for responding to many of them. So I'll start with questions that have been asking about sensitivity and specificity in people living with HIV and in children, if you looked at that within the Seq Treat project. Thanks.
So we did look at, I think the HIV results where they were available were taken in. So we did not, if I go back to that result, we did not actually see that significant difference unless it was correlated with lower bacterial load. With children, we did not. That was actually, it was above 16, was, I think, in fact, the inclusion criteria. Sadly, there are other groups working on specifically. I think always you want to make sure with in a way, the adult population to make sure a test works before you sort of take it out further. And that's always a challenge, right? You want to make it work for everyone, but at the same time, you don't know if it even will here. So, but that definitely groups like KNCVR and others are already interested in implementing this.
Yeah, and I would just say as well, our third session that will be on the 5th of June, you can address that question again to our Eswatini colleagues, because they have some work that they have done on stool samples So it'll be an interesting question to pose to them to see if, because it's from the Baylor group who seem to do a lot of work in children. So they will probably have some data that they can share. And then a follow-up question to your last slide where you were talking about whole genome sequencing from direct from sputum. And someone was asking, is DNA extraction performed on the raw sample, or do we need to culture the sample first? I'm not sure if the recovery will be sufficient, especially if it's a low bacterial load. Over.
Exactly. Actually, precisely to that point, that's why WGS cannot be done today yet directly from sample. For targeted sequencing, however, it has been shown possible because we are going to very specific targets. So even if I have all of the DNA in there in the sample, whatever I got, so there'll be TB there, there'll be other bacteria there, there'll be human there, but I am targeting very specific gene regions. And so that's what I'm amplifying, and hence for targeted sequencing that works. But you are seeing, of course, when there's very low bacterial load, that is making an impact, a negative impact on the ability to sequence. So any method, so that continues to be a reason. So right now, yes, for whole genome sequencing, we've not seen anyone effectively, other than a couple of papers that I've seen, to effectively get enough yield from just direct from a sample or a sediment to get there to do the whole genome.
Yeah, and then we also had a couple of questions around cost, and I know you addressed a few of them in your presentation, but perhaps if you could just speak to what are the key considerations for programs. Some people were really asking how can these approaches be adapted for implementation, and if you have an idea of the cost per test, if you're able to share.
Course, but I think there's some range given in the guidelines. I think the operational handbook, I think maybe talks about it. So I think considerations, again, this will be probably way better articulated in those guidelines in the operational handbook and the others than I can probably speak to right now. So that costing tool is one I would say. I think utilize some tool like that too, because then you don't have to think of all the components, at least to know all of the pieces that are needed, because I think that was one of the features we did. The price itself of the products may change or might be different also depending on the region, which we eventually don't want that as well. But right now there is that difference. But at least the components should all be there. Every person or every lab that's doing this should have access to all of those, or should know all of those pieces that are needed. So that would be a good one to, at least for planning purposes. I think the other one is then really that cost of, like, from a health program perspective, what is that spend, right? Because again, it's if you're talking from a programmatic sense, it's very much on that diagnostic line item, and within that, what can be done for drug resistance. So where countries have seen, again, I think your Eswatini and others, as you get countries to speak to this, I think that'll be very insightful, because that's where we've been trying to learn from countries. How do they make a case in a way, that investment case for sequencing? So right now, the fact that it's on Global Fund and other procurement lists and being updated there is important. So at least you have it at certain access pricing. Groups like Chai and others are working on obviously getting sort of that volume-based procurement, all of those pricing done. So that's at a larger global level, so that overall the prices should come to a reasonable level for affordability. But I think in terms of countries themselves for that investment case, I think that utilizing existing, is there ways to tap on an existing infrastructure? Is there core facilities that can be utilized for sequencing by multiple sites or labs, or a service method, or like a service lab way, because that will still be cheaper than setting up an entire sequencing facility. Now, again, this is all anecdotal. Different countries have told us about different things they've tried, which has worked very well. So there is no one answer, but I think there's 3 or 4 different ways to implement. So again, not every lab would need to set up sequencing from scratch, because that would then just be cost prohibitive at this time.
Yeah. No, thank you so much, Anita. And we really have a lot more questions than we can get to within this hour. And I just wondered if you had any last remarks that you can give in terms of what countries can do, and where you see really tNGS being placed potentially. Over.
Oh, that's a lot I can say. It'll be an entire hour. There's so much to ask. I would ask of the countries, of the folks on the call. But I think really to understand, I think the fact that you're interested, you're in this webinar, you're in the webinar series by ASLM is critical, right? Because you're seeing that need, on the ground. So what we need from you is, in a way, that advocacy and that push to your health programs, but also to, in a way, the global bodies to say, hey, what can you do to make it easier? And this is why we need it. I think you are the ones with the technical knowledge and with the clinical knowledge in that sense to say why this is important. But to kind of make that push, make that case, saying what is needed from you, saying, hey, either I need the guidance or I need access to more of these tools. I think hearing that from you is going to be the most important in terms of driving not just implementers doing some those specific things for you, but also for funders to know where they need to put the funding. I think that's always a piece where funders are always looking, okay, what is next? And now, with global health, with that sort of changes and everything happening, there are more actually other countries and other groups that are willing and interested in supporting. But I think to really be clear on what are those, some of those might be very fundamental things that you want addressed in your system. You may not need the latest, greatest sequencer. You may already have that. I'm actually very, very sure you actually have that in your countries already. But how do we link that to your TB program? How do we make sure TB samples can be sequenced? And that is that link that you need, which means some of that is those fundamental linkages in sample transport and so on. So I think very much of you have that voice now. I think that's all because this is that platform where I could ask that is like, you guys have the knowledge and the voice, but I think those gaps that you're seeing, this is in a way, I would really urge you all to make that known. I think there's some surveys that through Find WHO that had gone out. I think most of your, I think all your countries actually responded, which was just fantastic. That's going to give us a lot of intel in terms of what you're seeing, what are those gaps. I think any opportunity like that, and hoping with ASLM as well, to at least have systematically capture those so we can say, I think 50% of the countries say this is what is missing. I think that'll help us direct attention, investment, and actual action to tackle those so that you can utilize technologies like sequencing, not just sequencing, but really anything that makes a difference to your TB management and care.
Oh, thank you so much, Anita. And thank you everyone for joining. I'm sorry we're slightly beyond time, but thank you for staying on. And we look forward to seeing you again in our next session within this TB Genomics series. So have a good afternoon, good evening, good morning from wherever you're joining us from. Bye.